Okay, so welcome to another AP Bio video. We are going to continue talking about metabolism and free energy. So this little diagram is showing us the relationship between free energy, stability, and the capacity to do work, along with spontaneity. So we see in the top when our delta G is negative, we have a spontaneous process, something that happens without the input of energy. And with our negative delta G, we have a higher capacity to do work. Now, since we said this is a spontaneous system, it is eventually going to move to equilibrium. So once we are at equilibrium, we have less capacity to do work and less free energy. Lucky for us, our cells are not in equilibrium. They're open systems that are constantly experiencing a flow of materials in and out of themselves. A defining feature of life is that our metabolism is never at equilibrium. So an example that we can think about is cellular respiration. Essentially, as long as our cells have a steady supply of glucose or other fuels and oxygen, they can expel waste products and they'll never actually reach equilibrium and be able to continue doing this work of life. So an example of this that we can see is this little hydroelectric system. All of our little, what are they called? I don't know, water turbines. Our water turbines in an open system will never reach equilibrium, which is a lot like our cellular respiration. The sequence of reactions in cellular respiration is kept going by the energy differences between glucose and oxygen at the top of the hill and our carbon dioxide and water at the bottom of the hill. Now, before we go any further, I just want to check a very common misconception. So I know that I have used this language throughout the semester so far, and I know that, and I'm sure that you've probably heard this language in some of your other classes throughout your life. So what we're checking here is that energy is not actually stored in bonds, okay? Energy is stored in the molecules. Bonds are kind of an abstract idea, whenever we see like those ball and stick models, the stick doesn't actually exist in between molecules, in between atoms. It's more of a force. So there's no energy stored in that force. The energy is stored in the molecules. Whenever you hear energy stored in bonds, it's really shorthand for the potential energy that can be released when new bonds are formed and those original bonds break, okay? So our cellular respiration, when we're taking in glucose and breaking glucose apart into smaller molecules, what that is doing is freeing up energy to then be possessed by a molecule. And I'm sure you guys have heard this before, that the molecule that's possessed by energy in our bodies and in other organisms is ATP. So that's what we're going to talk about a lot going forward here is ATP. I linked a really cool article that talks about this misconception that energy is not actually stored in bonds, but energy is stored in the molecules. So check out that article. So ATP, we heard it's possessed by energy. And this energy in ATP powers cellular work by coupling or like pairing exergonic reactions and endergonic reactions. So reactions that are spontaneously happening and reactions that are not spontaneously happening. They couple those together to keep them moving on. And why do we need to keep them moving on? Because our cells have to perform work. So there's three main types of work that our cells perform. There's chemical work. So this is like rearranging monomers to polymers. There's transport work. We're pumping substances across the membrane, or we're pumping things against the direction of spontaneous movement, against their concentration gradients. So an example of that we know from last chapter or last unit is active transport. Our cells also perform mechanical work, like the contraction of muscle cells, or our little cilia's beating, or the chromosomes moving across the cell, like an anaphase of mitosis. we'll get on to, all into that later. I just absolutely love this animation. This is a kinesin motor protein that's moving a vesicle across one of our filaments. So cool. He's just really like boom, 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 just walking along. I love it. And all of that is powered by the coupling of endergonic and exergonic reactions by ATP. So we know that cells manage energy resources by using ATP to couple exergonic and endergonic reactions. But what does, ATP really look like? What is it? So ATP, the cell's energy shuttle, is composed of 3 parts. So we have our ribose, or a sugar, our adenine, and then 3 phosphate groups. So these phosphate groups can be broken by hydrolysis. When the terminal phosphate bond, or that last phosphate group, that 3rd phosphate group, is broken, then a molecule of phosphorus leaves ATP, which now it's no longer adenosine triphosphate, it's now adenosine diphosphate, ADP. Energy is released from ATP when that terminal phosphate bond is broken. Now, again, remember that this release of energy comes from the chemical change to a state of lower free energy, not from the phosphate bonds themselves. So the energy that is released whenever that phosphate group is lost is greater than the energy that most other molecules deliver. So that is why ATP is so useful to our cells and other cells for coupling these reactions. But why does it really release that much energy, or like so much more energy than other molecules? Well, all 3 of these phosphate groups are negatively charged. So there's like a mutual repulsion here. These negative charges are grouped together, which contributes to the great instability of this part of the ATP molecule. So that triphosphate tail, you can kind of think about being like a compressed spring. It is just ready to be uncompressed. It's ready to let go. I'm about to sneeze. Okay. And whenever that spring is let go, that 3rd phosphate group is lost, there is a release of energy. Now, there's a funny little comic here where our 3 phosphate groups are talking about how they love hanging out on this ATP molecule and that the bonds will never break and they'll be phosphate friends forever. That's really cute, but we kind of have to remember here that our phosphate groups are negatively charged, so there is a repulsion. You can kind of maybe read into our little phosphate groups' faces in the first part of the comic and saying maybe they are repulsed by that last one, and they're not that, distraught whenever he is lost and snapped away by hydrolysis. So yeah, cute comic, but you have to remember that all 3 of these phosphate groups are repulsed by each other. So how does this hydrolysis of ATP actually perform work? Well, the energy from the exergonic reaction of this hydrolysis is used to then drive an endergonic reaction later on. So the coupled reactions overall are exergonic, meaning they're going to happen spontaneously, but we have to have that original spontaneous reaction to drive the non-spontaneous reactions within our cells. And all of this happens by something called phosphorylation. So phosphorylation is just the name of transferring that phosphate group from ATP to some other molecule, like a reactant, in our next endergonic reaction. Whatever molecule receives that phosphate group is called our phosphorylated intermediate. But we know that ATP is a renewable resource. It can be regenerated by the addition of a phosphate group to that ADP. So it can cycle in between ADP and ATP. And the energy that is required to phosphorylate ADP comes from other catabolic reactions in the cell, from other spontaneous reactions in the cell. Again, we are seeing that energy coupling. So this ATP cycle is kind of like a revolving door through which energy can pass, and it's transferred from catabolic reactions to anabolic reactions. Just kind of some fun facts here. There you go. There's ELF in the revolving door, the ATP cycle. So just kind of some fun facts here about ATP and why we should be so thankful that it's renewable. A working muscle cell recycles all of its ATP in less than a minute. This is a really quick revolving door, okay? There's 10 million ATP molecules. There's a typo there, but 10 million molecules of ATP are consumed and regenerated each second. Again, really fast revolving door here. And if ATP could not be generated by adding that phosphate group to ADP, if we couldn't phosphorylate ADP, then humans would use up to our body weight of ATP each day. But all of this has to be carried out by enzymes. So they're spontaneous reactions, but they would take too long to happen. That revolving door would move too slowly for our cells to be able to do the work it needs to in the time that it needs to do it. So a catalyst is basically a chemical agent that speeds up the reaction, but it's not consumed by the reaction. And an enzyme is just a catalytic protein. So the example that we are looking at here is sucrase. Yeah. Sucrase. So sucrase is the enzyme that catalyzes the reaction of hydrolyzing sucrose. So we could put sucrose in sterile water and it'll sit there in that sterile water for years without undergoing hydrolysis to glucose and fructose. But if we add sucrase to that mixture, if we add the enzyme to the mixture, then it'll hydrolyze in seconds. So we can see how much these enzymes really do speed up these reactions. Now, another common misconception here is that enzymes are going to be changing the free energy, that the enzymes are going to be making an endergonic reaction exergonic. That's not actually true. We have to remember that enzymes are able to perform their function by lowering the activation energy. So let's talk more about this activation energy. So activation energy is just that initial energy needed to start the chemical reaction, okay? So every chemical reaction involves breaking bonds between molecules and creating bonds between molecules. In between that the original reactants and the final products, there is a really high unstable bond bending molecule, and we call that our transition state, okay? So changing one molecule to another involves that contorting the starting molecule into a really high unstable state before the reaction can proceed. So this contortion is kind of like bending the key ring. It's kind of like bending the key ring. So the key ring is highly unstable in its open form, but returns to stability in its final form. And without the work of enzymes, our chemical reactions would be too difficult. So yes, Thomas Fuller said this, but I think an enzyme would also say the same thing, that all things are difficult before they are easy. Our enzymes lower the activation energy of reactions, allowing the reaction to continue easier than it would be without it. So let's talk more about enzymes. We know that our enzymes are specific. There's a specific substrate that can be bound to that enzyme. Whenever those are bound together, we call that the enzyme substrate complex. And the enzyme, once it is bound, is able to convert that substrate to a new product.
Watch how substrate binding to an enzyme active site induces a change in the tertiary structure of the enzyme. These images were created using X-ray diffraction data of hexokinase with and without bound glucose. Notice how glucose binding to hexokinase causes a conformational change in the enzyme that closes the active site. Credit: Mark B. Gerstein, Yale University.
So that specificity is what determines which chemical process will be happening at any given time. Whenever the substrate binds to the active site, something happens that we call induced fit. So induced fit is whenever the chemical groups of the active site are kind of brought into positions that kind of hug that substrate, enhancing the ability to catalyze the reaction. So a few of our amino acid groups are able to kind of hug or have that induced fit around our substrate, which we are seeing here in this little animation of an enzyme. So the enzyme that we see here is called hexokinase, and it's going back and forth between those two shapes because the substrate is bound or whenever the substrate is not bound. And a lot of people call this enzyme dancing. So here's kind of a funny picture that was included in this research article talking about choreographing an enzyme's dance. So the dancing enzyme is going between its two shapes in an active form and an inactive form according to whether or not the substrate is bound. So only a few amino acid R groups will actually convert, do the converting from the reactant to the product. The rest of the amino acids exist only as structure. But we have to remember that they're really fast acting, and not only are they fast acting, but they are reusable. They're not actually consumed by the reaction. And because of this, small amounts of enzymes can have huge metabolic effects because they can be repeatedly used. So this is kind of just a little animation or graphic that shows us substrates entering the active site, creating the enzyme substrate complex. The activation energy is then lowered, and then the enzyme, where a few R groups of amino acids of that active site are able to convert these substrates to products. Then eventually, once they are transformed or converted, then they are able to be released. And now the active site is available for new substrates to enter, and the cycle continues. But how does the active site actually lower this energy barrier? How does this third part of this reaction take place? Well, it can happen in 4 ways. So one is orienting the substrates correctly, making sure they're fitting into that specific position. We can be straining the substrate bonds, so that induced fit helps us kind of contort those bonds into that transition state. The active sites also might provide a favorable microenvironment. So maybe the substrate is extra acidic, and that little pocket, that little active site might be an acidic section in an otherwise neutral enzyme. Or our active site may actually covalently bond to the substrate. So here's a little bit more information about each of those. These slides will be online for you guys to look into more yourselves. Now we know that enzymes aren't always recyclable. Sometimes enzymes are denatured, and they're denatured whenever there's environmental conditions that are outside of their optimal conditions. So our optimal conditions can include things like a certain temperature range or a certain pH range, and once we're outside of those conditions, then the enzyme might denature. So here's kind of a cute little comic that reminds us of this idea by talking about how enzymes would be such fussy or annoying coffee customers because they have such specific conditions that are needed. Now, we said that there's a certain temperature range that enzymes are able to still perform their work, but so up to a point, an increasing temperature will increase the rate of reaction. It'll actually make the reaction happen faster. But after that point, then the enzyme will become denatured and will be no longer able to perform work. I don't know if y'all can tell, that little guy at the bottom is supposed to be a thermometer. He's supposed to represent temperature. And the enzyme and the substrate here are saying we're best friends, nothing could ever denature us. But then the little temperature guy says, I can denature you and so can my friend pH. So let's talk about cofactors. Cofactors are non-protein enzyme helpers because enzymes and proteins can do a lot, but they can't do everything. Sometimes they need help from one of these cofactors and able to do its job of transforming the reactants to the products. So these cofactors could be bound tightly and permanently to the enzyme, or they may be bound loosely and reversibly along with the substrate. So they may come on and come off along with the substrate. So there's some cofactors that are inorganic, such as a metal, like, for example, iron. Y'all know I can't say that word. Iron, an iron ion. Or these cofactors could be organic. And if they're organic, they are called coenzymes. There's lots of different types of coenzymes, some of which we will talk about, but just know that coenzymes include vitamins. So this animation is showing us an example of a cofactor that is binded loosely and reversibly along with the substrate. So you see that cofactor is needed to come onto the enzyme along with the substrate, and then once it's there, it helps the enzyme turn that original product into the final reactants. Now we have cofactors, things that help the enzyme do their job, but we also have enzyme inhibitors. So there's two types of enzyme inhibitors. One is a competitive inhibitor, which binds to the active site of the enzyme, which we can see in this comic over here. So that competitive inhibitor is now bound to the active site, and the substrate that it's meant for that active site can no longer bind because it's in the way. We also have non-competitive inhibitors that bind to a different part of the enzyme. So this causes the enzyme to change shape. We're changing the amino acid sequence, and it's then changing the tertiary structure. It's changing its shape and making the active site less active. So it makes the active site less able to bind to its desired substrate.